Denaturation and renaturation of DNA helix. Denaturation of DNA is a loss of biological activity and is accompanied by cleavage of hydrogen bonds holding the complementary sequence of nucleotides together. This results in a separation of the double helix into the two constituent polynucleotide chains. In it, the firm helical two-stranded native structures of DNA is converted into a flexible single-stranded denatured state. The splitting of DNA molecule into its two strands or chains during denaturation is obvious because of the fact that hydrogen bonds holding this bases are weaker than the bonds holding this bases to the sugar phosphate groups. The transition from native to denatured form is usually very abrupt and is accelerated by reagents such as urea and formaldehyde, which enhance the aqueous solubility of the purine and pyrimidine groups. Denaturation involves the following changes: increase in absorption of ultraviolet light, increase in absorption of ultraviolet light, increase in absorption of ultraviolet light, hyperchromic effect. As a result of resonance, increase in absorption of ultraviolet light, that is the hyperchromic effect. Hyperchromic effect. As a result of resonance, all of the bases in nucleic acids absorb ultraviolet light, and all nucleic acids are characterized by a maximum absorption of UV light by wavelengths near 260 nm. And the native DNA, which has base pairs stacked similar to a stack of coins, is denatured. There occurs a marked increase in spectral absorbency of UV light by pyrimidine and purine bases, an effect called hyperchromicity. Hyperchromicity or hyperchromism, which is due to unstacking of the base pairs. This change reflects a decrease in hydrogen bonding. Hyperchromicity is observed not only with DNA but with other nucleic acids and with many synthetic polynucleotides which also possess a hydrogen bonded structure. Then decrease in specific optical rotation. Native DNA exhibits a strong positive rotation which is highly decreased upon denaturation. This challenge: decrease in specific optical rotation. Native DNA exhibits a strong positive rotation which is highly decreased upon denaturation. This change is analogous to the change in rotation observed when the protein is denatured. Decrease in viscosity. The solution of native DNA possesses a high viscosity because of the relatively rigid double helical structure and long rod-like character of DNA. Disruption of hydrogen bonds causes a marked decrease in viscosity. Effect of pH on denaturation. Denaturation of DNA helix also occurs at acidic and alkaline pH values at which ionic changes of the substituents on the purine and pyrimidine bases can occur. In acid solutions near pH 2 to 3, at which amino groups bind proteins, the DNA helix is disrupted. Similarly, in alkaline solutions near pH 12, the enolic hydroxyl group ionizes. This prevents the keto-amino group hydrogen bonding. The absorbance spectra of DNA solution at 260 nanometer and pH 7. When mixtures of nucleotides are present, the wavelength at 260 nm dashed vertical line is used for measurement. 260 nm is very important. Then the absorbance relative to that of 25 degrees Celsius is plotted against temperature. The wavelength of the incident light was 260 nm. The TM about temperature is about 72 degrees Celsius for Escherichia coli DNA and 79 degrees Celsius for the bacterium Pseudomonas aeruginosa DNA. 66% GC pairs. Effect of temperature on denaturation. The DNA double helix, although stabilized by hydrogen bonding, can be denatured by heat by adding acid or alkali to ionize its base. The unwinding of the double helix is called melting because it occurs abruptly at a certain characteristic temperature called denaturation temperature or melting temperature. The melting temperature is defined as the temperature at which half the helical structure is lost. The abruptness of The transition indicates that the DNA double helix is highly cooperative structure held together by many reinforcing bonds. It is stabilized by stacking of bases as well as by pairing. The melting of DNA is readily monitored by measuring its absorbance of light at wavelength near 260 nanometer. As already stated, the unstacking of the base pairing results in increased absorbance, a phenomenon termed hyperchromicity. TM is analogous to the melting point of a crystal. It can be lowered by the addition of urea, which disrupts hydrogen bonds. In 8 molar urea, TM is decreased by nearly 20 degrees Celsius. DNA can be completely denatured, separated into a single stranded structure by 95% formamide at room temperature only. Since the GC base pair has a 3 hydrogen bonds compared to 2 480, it follows that DNAs with high concentrations of G and C might be more stable and have a higher TM than those with high concentrations of A and T. In other words, the DNA molecules containing less GC bond denatures first, and GC bond has higher thermal stability. In fact, the TM of DNA from many species varies linearly with GC content, giving from 77 to 100% of the fraction of GC pairs increases from 20 to 78%. This measurement may henceforth be used as an index for heterogeneity of nucleic acid molecules. For instance, viral DNA has a much sharper thermal transition than that prepared from animal sources. Complete rupture of the two-stranded helix by heating is not a readily reversible process. However, if a solution of denatured DNA prepared by heating is cooled slowly to room temperature, some amount of DNA is renatured. Maximum reversibility is usually attained by annealing, slow cooling. The denatured DNA, holding the solution at a temperature about 25 degrees Celsius below Tm and above a concentration of 0.4 m molar Na plus for several hours. Fast cooling will not reverse denaturation, but if the cooled solution is again heated and then cooled slowly, renaturation takes place. The restoration occurs because the complementary base reunite by hydrogen bonds and the double helix again forms. Renaturation of complementary angle strands to produce fully double-stranded molecules requires two separate reactions. Native DNA, we provide heat, then denatured DNA is formed, nucleation is giving second order, then zippering is happening. So then forms the renatured DNA. Steps in the denaturation and renaturation of DNA fragments. Nucleation reaction. In this, hydrogen bonds form between two complementary single strands. It is a biomolecular second order reaction. Zippering reaction. In this, hydrogen bonds form between all the bases of the complementary strands. It is a unimolecular first order reaction. Molecular weight of DNA. DNA molecules are among the largest known. It is difficult to isolate DNA without fragmentation. The entire DNA of bacteria such as Escherichia coli has a molecular weight of 2.6 into 10 raised to 9. Viral DNAs range in size from about 1 to 350 into 10 raised to 6. The residue weight of a single nucleotide is 300 to 350. Thus, there are about 3000 nucleotides per million molecular weight of DNA. Length of DNA molecule. The amount of DNA is usually measured by the microunit of the weight known as the picogram (pg). The amount of DNA has been found to be constant from cell to cell, and 1 picogram is equal to 10 raised to minus 12 gram. 1 pg is equal to 1 into 10 raised to minus 12 gram. A piece of double helical DNA, 31 cm length, weighs 1 picogram. So, by taking the weight of DNA amount in the nucleus, the length of the DNA molecule can be calculated easily. By employing this method, scientists have calculated that the human diploid cells contain 5.6 pg of DNA amount, which is equivalent to 173.6 centimeter long DNA molecule. Similarly, they also calculated that a diploid cell of Trillium has dsDNA of 37.2 meter length, which is equivalent to 120 pgDNA by weight. Also, the polytonic chromosome of Drosophila with 293 pgDNA contents have 90.83 meter long DNA molecules. Shape and size of DNA molecule. The DNA molecule are characteristically highly elongated structures. The Escherichia coli chromosome is a single molecule of double helical DNA containing about 4 million base pairs. This DNA is highly asymmetric. Its contour length is 14 into 10 raised to 6 angstrom and this diameter is 20 angstrom. Whereas this contour length 1.4 mm corresponds to a macroscopic dimension, its width 20 degrees Celsius, 20 angstrom in the atomic scale. However, the largest chromosome of Drosophila melanogaster contains a single DNA molecule with 6.2 into 10 raised to 7 base pairs. These have a contour length of 2.1 centimeter. Such highly asymmetric DNA molecules are very susceptible to fragmentation by shearing forces.